Okay, I will begin by welcoming everyone to this CyVerse webinar on transcriptomics with non-model organisms by Blake Joyce. I'm Martha Naro, and I coordinate most of these webinars. I will now let Blake introduce himself and begin the webinar.
Hey, good morning, everybody, or afternoon, or whatever time zone you're in. Welcome. This is my second webinar. We'll be talking about non-model comparative transcriptomics. And as I was saying before, we're going to try to go through each of the steps pretty systematically. And so we may not get through the entire analysis because there is quite a lot to see and quite a lot to learn and do. And I have a little bit of an intro, but it'll be mostly a live demo. And so I will try to get to that as quickly as possible. And then, of course, please stop me at any time and ask questions. If anything at all is unclear, or if you have a different kind of analysis in mind, or if you are interested in other options at any given time, please don't hesitate to ask. I have been doing this for some time, but just like any science, I don't know everything. So if there's anything that I don't know off the top of my head, I will be happy to look up and then email you and follow up with you after the webinar is over. So I will just start with a quick note and say that my background is not bioinformatics. In fact, I am a life scientist, like I assume most of you are. I began as an ecology student at the University of Georgia, and then I became a molecular biology and biotechnologist, mostly secondary plant metabolism engineering, for my master's and my PhD. And so I've come to learn a lot of these techniques, much like I would imagine most life scientists would have to. And that is, I had some interesting questions that I couldn't answer in any other way than to do the big data and metabolomics kind of analyses. And that led me very quickly to transcriptomics. So my initial work was actually in soybeans. I was working with Wayne Parrott at the University of Georgia. I inserted one gene, a phytoene synthase, into soybean, and voila, it makes beta carotene, which turns the soybean orange, which is kind of fun, of course, to put on a poster. And that was a pretty fun thing. But then you get into these non-model organisms like this plant. This is Pittosporum resiniferum. In which case, that actually makes gasoline, proper gasoline. And so the problem with that is that it's a non-model organism, it's an exotic plant, it's never been studied genetically, but it has very fascinating traits, and that is to say, literally biologically derived gasoline. Those are just fruits that are straight off the tree, burning that gasoline. We had quite a lot of fun with that. And so I very quickly got into having to figure out non-model transcriptomics so that we could rapidly get into asking questions about these metabolic pathways. And so these days, a lot of people are starting to do similar work with not only plants, but many, many different species. And we're starting to move away from the model organisms. So to that, I'm going to teach this webinar, and I certainly will follow up any questions, basically kind of in a couple of different ways. First off, I will try to explain what I have found to be best practices. I find I teach what I wish how I had learned rather than the process that I learned. I did a lot of failing in the early years of doing bioinformatics and learning the hard way. And so I will try to skip that bit and get you to the things that I find very important and useful, and just things like organization that'll help keep your day-to-day easier when you're going through, because it can be a very complex process. This is going to be a very brief introduction to de novo transcriptomics. There are many, many possibilities. There are many more tools, more being developed daily. We'll be using the Trinity and the Trinity package in this webinar, but that is not to say that there aren't many, many other great tools that are available. And so what I would like to say is that this is going to require a bit of experimentation, just like any science. And so I'm going to also hopefully teach you a few techniques on the computational science world side of bioinformatics that hopefully will help you get comfortable with computation, get comfortable with executing in different environments, and also get comfortable with leveraging some of the CyVerse resources. But also it may help you leverage some of your local resources that are available to you, and teach, and generally move you towards being a little bit more of a bioinformaticist. So we're going to look at some things like the best practices, as I said. We will be doing some command line execution. I remember as a beginning bioinformatician that the command line was incredibly scary to me. And so we'll go through that very slowly. I'll introduce some of those, and we have quite a lot of documentation to that effect that can help for follow-up. But I highly advise taking some notes on some of the commands that I'll execute, because these are all incredibly useful ways of talking to your computer or many other kinds of computers. And probably the biggest thing that I will be showing you is how to move data pretty fast, but really getting the idea of local compute, which is the computer you're sitting at, versus remote compute. And that is to say, computers that are at your HPC, computers that are at national resources like Exceed, or national resources and international resources like CyVerse. And so there are ways to actually have computer-to-computer communication, but it can be a little confusing about where you are from time to time. So we'll work. I'll try to explain at each step exactly where we are and kind of bridge those two things, because we'll be using cloud compute, we'll be using HPC through the discovery environment, and I'll be doing everything from my local laptop. And so just a few things very important that I'd like to mention as well. And this is something that I had learned as a beginning bioinformatician as well. I see our job here at CyVerse very much is to minimize the things that are not useful for science, or for your science, as the case may be. And that is to try to minimize how much time you spend installing packages, or dealing with dependencies for each of the software packages, or running make files, or doing all these things that are not science. But the caveat there is all the time that you save by not having to do installations or figure some of these things out, you really do have to spend on understanding the bioinformatic tools, understanding the science behind the tools, understanding that there will be failures, and we may even see some today. And so one of the things that you can really learn very quickly, and it's very important, is how to diagnose failure, how to seek help, and I'll show you some of those resources that we have. And in addition, just to understand that bioinformatics is absolutely a science. And so when you're working with your organism, whatever organism that may be, you may have to run a few different kinds of pipelines and run a few different kinds of assessments, and then look at how it all comes out, and then decide, this I think is the best of the 3 or 4 things that I've tried, and then to move forward each step that way. Very frequently, and of course when I started as well, I ran through an entire pipeline and I thought I was done, and I was just so happy to be done that by the time I actually got to the bench and started doing things to confirm my bioinformatics findings, it turns out that I had not assessed that one step, and it had thrown everything off. And so I will show you a lot of the assessment tools that we have, and that's a bit different than a lot of the pipelines that I've used in the past and certainly have seen around. I am a firm believer in assessing at each step and understanding your data and understanding your outputs. And then when you're comfortable with what they are, you move forward to the next step. And just like any other kind of science, right? You don't run a gel or run a PCR and then just assume it works. You always have to verify. So anyway, with that, I will move on. So just a quick overview of the local versus the remote. We're going to be using the discovery environment, which is our graphical user interface, where there are a lot of apps that are available. There's a lot of point-and-click apps, and that's very great, especially for beginning scientists. You can get in, you can play around with each of these tools, you can run example data otherwise, but it exists elsewhere. So I'm going to be using a laptop, and that laptop currently is located in Tucson, Arizona. But we're going to speak out to the discovery environment. And I'll show you how to move data from your local computers out to the discovery environment. But we'll also be using the discovery environment to talk to Atmosphere. And underneath the discovery environment is what we call the data store. And so that's where your data goes. Every user who signs up, sign up is free. Every user gets 100 gigabytes, I apologize. as space. If you need more space, you can request it. It's a simple form. And what's nice about the data store is once your data is in that data store, operating between the discovery environment, Atmosphere, and many of the other platforms in CyVerse is incredibly fast. And should something happen locally, that is to say, here's our wildfires from last year, that's Tucson mountains burning. Even if you're cut off locally, you can still access your data that's in the data store, keep running analyses, keep moving forward with your science. Or say, like me, if you tend to wander off into the world, because your data is in the data store underneath the discovery environment, you can actually continue running analyses, continue doing science, and all you have to do is you're going to remote in locally, and all you need is internet, basically. And so I was doing this when this is Lashan, China, so I was actually doing work while I was on vacation and doing other things. And so it's a very nice way to continue, because life is a little hectic, and as scientists we tend to travel a lot and have a lot of other things going on. So I'm a big fan of having the compute working kind of around the clock if you can possibly do that. So today I will be showing you how to access the discovery environment through the browser. And I'll also show you how to SSH into a cloud computer. And that's going to be through our Atmosphere platform, CyVerse Atmosphere platform. There's also ways to access Atmosphere through your browser, and I'll show you that as well. But basically, once things are in there, you can work on them from basically anywhere, as long as there's an internet connection. So without further ado, we will get down to the live demo. So I'm going to go to the CyVerse.org website. You can register or sign in up here. I'm going to slide down just slightly and go to the discovery environments. It's right here. This is the splash page. There's lots of details here about the discovery environment. And let me minimize. Excellent. And so when you actually launch that, it's going to take you to a sign-in page. I'm going to assume, I've already signed in, and so it's going to go ahead and open up. I'm going to assume that everybody has registered for CyVerse. If not, it's pretty easy to do, and we have a link there. And if you go here to the sign-in page at the very top, you can register as a new user here at any time. So inside the discovery environment, it starts off looking like this. You'll notice that there's a data tab. That's your window into the data store. There's going to be an apps tab. That's where all of our analytical tools exist. And then there's an analysis tab. And so that's a very quick way to get at any of the analyses that you've actually run. So we'll open up the data tab. For this tutorial, for this webinar, I'm going to be using a few of the Trinity and the Trinity example data. I have a few other kinds of example data that I will also show you, because there are other analyses that I did that were interesting, and we'll go through that. But actually, all of this is available to you as a community member here in this tab underneath community data. This will say your username. So my username is bjoyce3. If you ever want to share something with me, that is where you'll do it. Underneath here in the community data folder, you can click on that. You'll see lots of different kinds of data that are available to the public, that is you or otherwise. If you click on the Trinity transdecoder Trinitate, I have a lot of different kinds of example data here. And again, today we're going to be working on the Jove non-model de novo transcriptomics example data. I will also mention that we recently published this protocol in the Journal of Visualized Experiments, and that's where the Jove part comes from. And so there's actually a video protocol for each of these steps as well, in addition to this webinar. And so that's why I would love for the webinar to be interactive, and that you would stop me and ask questions, because I can go back and improve documentation, add in things that I had not considered, or respond to each of your questions in some way or another. So again, please stop me at any time. So within that example data, I have a number of different types of data. I'm a plant guy, so I tend to think plants, but I have some animal examples in here, some invertebrate animals. I have some fungal examples in here, and we're going to be working in the official Trinity data set. Now, the very first thing I'll say as far as best practices is that I really like to organize my folders. And I like to organize them in chronological order of how I started, how I did the analyses, and every step thereof. I do that for two reasons. First, my future self thanks me for being slightly organized. 6 months from now, when I've forgotten this webinar, yes, that's a great question. I will link out to that publication. It should be published within the official link should be out there in the next week or two, and I'll be sure to follow up. So for the organization, the very first thing I like to do is have my raw sequences. In this case, these are the Trinity raw sequences they provide inside of their packages. They are named very simply left reads and right reads, and they are FASTQ files. So this is paired in data. There's left and right. There's a couple of different ways of saying left and right, but each sequencing platform seems to have its own way of doing things. But I like to consider this as an entire project, and that way, if I order them, I know exactly what step I did everything. I can very easily go back to any of the apps that I ran, and I can give this to anyone else, my colleagues, my collaborators, my reviewers. 6 months from now, when my paper is finally reviewed and getting close to acceptance, I can actually share all of that data, all the raw data, all of the analyses, all the versions of all the apps very easily with those reviewers. And they can very simply go down and recreate each of my analyses, and they can see for themselves how I derived my science. I find that very powerful for reviewer one who always wants to know all that stuff. I don't know why it's always reviewer one that wants to know that stuff, but it seems to always be reviewer one. So, and we'll move through as we go. I've gone ahead and added a few things just in the sake of time. I'm going to run this webinar a little like Julia Childs, and that is to show you how I put things together. And then I'm going to put the turkey into the oven, and voila, things will come out of the oven immediately cooked. But of course, you and I understand that in the real world, when you submit analyses, they can run for hours or days. In the case of assemblers and annotation especially, it can sometimes run for weeks. The pipeline that we've put together, you'll be able to see that and keep an eye on your analysis and make sure that it's running and know when it's going to return to you. And I always like that a lot. So we'll go through each of these, but I've already gone ahead and populated some of this stuff because, in the sake of interest, I was trying to save some time. So that's the general overview, and this is the data tab, and I'm going to be operating out of here. So every time I complete an analysis, my ultimate goal is to get it back into these folders so that I can have a very organized project structure, and we'll work all the way through, and at the end we'll have that. Now, the very first thing that we're going to do is we're going to take these raw sequences from this folder, and we're going to assemble them in Trinity. Trinity exists in the DE as an app, and Upendra, my colleague as a science informatician, is actually updating that app as well. That should be out in the next month or two. It's the most recent version of Trinity that they're putting, and then he's putting it together through what we call Agave. That is our science APIs, and it basically allows us to leverage very large compute. The DE is an HPC as well, technically, and it runs a number of cores, but we can also reach out to national computing resources through Agave, and we can run it on many, many cores with lots of memory. And so for assemblers, that tends to be the way to go. So when we do our follow-up emails, I will also update our documentation when that app comes out. And at some point, you'll be able to run nearly the entire pipeline through the DE interface. But for now, we'll go over to Atmosphere.
Blake,
so this is, yes.
Sorry, let me break in and have people answer in chat. Can they see the labels on the folders clearly enough, or do you need to increase the font?
Oh, yes. Yeah. How is my resolution? Do I need to increase it? I probably should.
So let's do a yes, no. Can you see what he's doing okay? Or just bump it up one. That's
okay. Yes, thank you. Yeah, if at any time you can't, please don't hesitate. Yes, thank you.
Yeah, okay. I'll be quiet again.
Okay. Is that better? Anyone who couldn't see it before still can't see it.
I think that's a bit better, and will show up
Excellent.
better in the recording. So, yeah.
Okay. I will also try to I'll try to be mindful of that as we go through. So this is the data store. So this is actually not on my computer. I'm accessing the data store through my browser, right? Here's my local computer. Here's Tucson. This is outside. This is remote. Likewise, we have our cloud computing resource, which is also a remote resource. So this is Atmosphere. To get here, all I have to do is go back to the CyVerse homepage. I'm going to go back to CyVerse.org. And if I click on Atmosphere, you have a lot of information here. As a first-time user, it's sometimes good to read this. I'm going to go in and go ahead and launch Atmosphere. And because I've already signed in once, it already knows that I am me, and it will register everything. And this is the interface that we'll be using for talking to Atmosphere. At the very beginning of it, I will just mention that Atmosphere runs a little bit differently as a cloud computing resource than the Discovery environment does. And that is to say, every month you get a certain number of hours. And that is calculated by how many cores, how many CPUs you have running, times how many wall hours, how many real-time 24-hour-a-day wall hours. So all of that information is provided to you right up front. Right here in your allocation, it'll tell you how many more of those allocation units, how many more of those hours you have to run. You can see I've used 74 this month. And at the end of March, on April 1st, which is next Saturday, I believe, all of this will reset for me, and I will get another allocation. If you ever need more, all you have to do is press this button, need more, and you just fill out a very simple, I need this amount of resources. Maybe you need more CPUs, maybe you need more memory, or maybe you just need more allocation units, more hours to run. And we just ask you for reporting purposes, what is it you're doing? So in this case, you would write de novo transcriptomics with Trinity and Trinity. Just because we like to know which of our apps require more allocation units, so we can be sort of mindful of that. And it's also for reporting. We all have grants to report on regularly. So there's a lot of information here, but what I'm going to take you to is the projects tab. I've set up a project here. You can create new ones as well. And within that project, you can actually have lots of different things going on. So what's really nice about cloud computing is you literally ask for a computer. That computer exists in the cloud, in the ether. I like to call them ghost computers because that's a little bit closer to me. Like they kind of wink into existence, and when you're done with them, they wink out of existence. And in this case, I actually have 3 different computers running. Each of them are running a different version of Trinity. So what I can actually do is, let's say I have, I don't know, 5, or let's say 6, just for argument's sake, 6 treatments. So I have 6 different sequencing libraries. I can actually set each one of these up to have treatment 1, treatment 2, treatment 3, etc. You would probably want to name them appropriately as well. And each of these run separately of each other. And I can run 3 or 6 different assemblies at any given time. And what's really nice about cloud computing is I can just let them run and forget about it. I don't have to worry about somebody tripping over the wire of my local computer in my lab. I don't have to worry about someone else trying to run an assembly and crashing that computer, which has happened to me actually. I don't have to worry about the janitorial staff turning off the switch breakers for the holidays, which has also happened to me. I don't have to worry about any of that stuff because this exists elsewhere. And so that's really nice. And so this is one of many ways to get quite a lot of compute done in a fairly short amount of time, which always seems to be something of interest for people. Now, we're going to be using this instance.
A question, Blake,
yes.
Not in the chat.
Yes, you can absolutely do assembly without Atmosphere. You can do the assemblies here in the Discovery environment. As I was saying before, here's the Apps tab. I'll go ahead and show you this real quick. We're going to, let's see, Trinity, why not? So as I was saying before, Trinity does exist inside of the Discovery environment. We're in the process of updating to the newest version. And so that's why I'm showing you the Atmosphere. But also the Atmosphere platform is really, really useful. And so these are absolutely available as well. And I can even show them to you. In which case you just give inputs and parameters. We'll be running tools in the discovery environment, and I'll show you how to do that with some of the other tools. And when Apindra finishes the newest version of Trinity, again, that will be available to everybody. And I will certainly update everybody that's on this webinar. Is running an experiment on Atmosphere platform free? Yes. We're NSF funded, so everything's free if you're an academic. For industry, that's not the case, but we have worked with industry partners before, and we basically wind up charging just for the compute resources that is used. There's a federal grant rule that says that academics can't fund or can't subsidize industrial work, of course. Could you show us how to start a project? Yes, absolutely. And I will show you that in just a second. Government. Yes, I believe government is okay. Is that correct, Martha? NSF? Yeah, absolutely. Yeah. So government's fine. It's just industry.
Yeah, USDA. That's fine.
Excellent. Yeah, that was my understanding as well. Yeah. So yeah, academics and government is free. So the one thing I will say that for Atmosphere, and again, this is in the wiki, and that's available in the email and in the announcement for the webinar. But I will show you the wiki. But to actually register for Atmosphere, all you have to do is you sign in, and this is all spelled out in great detail. But when you log in here, this actually takes you to the user portal. So I'll just go ahead and show you guys this. And right here, instead of go to Atmosphere, it'll say register for Atmosphere. And you just put in your name, your institutional email. You tell us, what kind of cool stuff you're doing. It doesn't have to be very specific, just kind of broadly for reporting purposes again. And then, you'll get granted access. Usually it's within 24 or 48 hours. Oftentimes it's much faster. And then you'll have access to Atmosphere. You automatically, when you sign up for CyVerse, you automatically have access to the discovery environment. That's just granted to you right off the gate. Not everyone uses cloud computing, but for everybody in this webinar, you will most likely wind up using cloud computing. And so I would suggest going ahead and registering for Atmosphere. Let's see. I'm going to show you guys how to start a project. And yes, great question. So you can technically launch as many instances, as many of these cloud computers as you want. There's only 2 or so limitations, 3 if you want to think of it this way. First, you're limited on the maximum number of CPUs, that is cores, that you can have. So I, for instance, here am using 4 of 16 in the iPlant cloud in Tucson. I also, as an instructor, like say for this course and this webinar, I also have access to the iPlant workshop cloud. So if you're teaching a class and you want to have 30 students all working on Atmosphere for doing Trinity or doing Trinitate or whatever you'd like, you can actually register as a professor or as a teacher for access to the workshop cloud. And so on that, I have 32 cores that are available to me, and I'm using 13 of those for this demonstration. So you're limited on the maximum number of CPUs. And again, you can request more, of course. And you're also limited on the maximum number of memory, or the maximum amount of memory. In this case, on Tucson, I have 128 gigabytes of memory total. And I can distribute, what's interesting though, what's fun, is I can actually choose to distribute that however I please. I can have 16 individual computers that have 1 core, 1 CPU, and that's totally fine. Or I can have 2 computers that have 8 cores each. If you need more than that, again, you can request resources. But there's also the big brother of Atmosphere that was released last year, and it's called Jetstream. Now there you can ask for very large compute. In which case, if you're interested in that, please don't hesitate to contact me, and I will certainly send you documentations about Jetstream and how to move it out to Jetstream. I use Atmosphere routinely for doing Trinity and Trinitate, but sometimes I find it useful to move out to bigger compute, especially for bigger projects. Does that answer? Hopefully, I believe that's all the questions except for the projects. We're going to do the projects. And then, of course, I will also mention that the other limitation is how many of those allocation units you have. So as you have these cores open and running, it's burning these allocations for every real-time hour times this number. So in my case, I'm going to be burning 4 allocation units on the Tucson cloud for every real-time hour. So that's 4 times 24 hours a day for an entire month. And I usually factor 31 days because some months you have 31 days. And again, it'll reset every month. And so you get these allocations back every month. These are maximum. So once you use that 16 cores, you have to request more. Those don't reset every month. It's just as you're running. Now, let's get back to the projects. I will show you how to create projects. And it is simple as, here's my project. I'm going to do X, Y, Z, etc. I like to be incredibly descriptive, preferably painfully descriptive. Now, one of the other things I'll also say as far as best practices, try never to use special characters. And again, that's in the wiki and certainly in the protocol. But special characters are things like exclamation marks, hashtags, at signs, money, dollars, right? Percentage signs, these sort of things. It's just best not to use those if you can. And that goes for naming files, naming projects, naming basically anything. Because in the computational world, sometimes the special characters have meaning. And so if you put those into your files or your folders or into your data or otherwise, it can cause a lot of trouble, and it can sometimes be hard to figure out what that trouble is. So that's really a tip that I learned first thing. When it comes to file names, spaces and special characters are just the worst. You don't want to have them. Yes, a space, especially in file names, spaces are special characters. And so I'll show you in a little bit, but if you have a space in a file name, when you're actually trying to transfer data using the terminal or using bash, that will cause all kinds of trouble. So you'll notice here that I've done underscores. So my suggestion would be, if you're going to do a space, do an underscore, or sometimes people like to do what we call camel caps, and that is to say they look like camel humps, right? Here's the capital of the first letter, capital of the next letter or word, capital of the next word. And you do these camel caps for each of the words. You just get rid of the spaces altogether. Great question. Thank you, Meg. So I'm just going to press create, and voila, here's my project. And it's blank, and you can add anything you'd like to it. Now, I will also show you that you can actually associate images that you like. You can create volumes. So this is actually a really great way to store data and transfer it between different cloud or different running instances, cloud computers. It works a little bit like a USB stick. You can literally, but it's in the cloud, and you can literally move data like a USB stick between different kinds of computers if you like. And that's generally the project. There's lots of, yes, thank you, Tanya. So instances are running cloud computers. They are an instance of a cloud computer. We sometimes call them virtual machines because, again, these are actual computers. They just don't physically exist anywhere that you would see them. They exist on a server somewhere in a basement in a cool room. To answer Randy's question, yes, absolutely. And I will show you how you can move data out of the DE. And we have a number of different ways. I'm going to show you the command line version, but we also have graphical user interface, drag and drop, point and click kind of things for moving data into Atmosphere. Yes, that's all the questions. So volumes are, again, those attached data storage that you can move very easily between different instances. And images are actually just software. So literally, it is a picture of a computer. It's a picture of all of the software that is on that computer. And so it is literally, so that's why we call them images. And so when you want to start a new instance, when you want to create a new computer, what you're going to do is go in here. You're going to look up Trinity or Trinity. And then you're going to find that software that you want. I've actually linked out to these images inside of the documentation, so you can very easily click on that URL and just go straight to the image that we're going to be using. But for other people who are just following along, we're going to be using this Trinity RNA-seq annotation. This contains Trinity and Trinity. And so what we've basically done is we've selected an image. These are things that we build or community members build. You create a computer, you install a bunch of software, and then you take a picture of all that software. The hardware goes away to be used by other people, but all of that information about the software and the installation and the date and the versions are saved forever as a picture, right, as an image. And all you have to do to create a new computer is to tell it what software you want, tell it what cloud you want to be on, and then tell it how much of these resources you'd like. I often, for Trinity and Trinity, I recommend not having anything less than 4 CPUs. In this day and age, 4 CPUs is pretty normal to have. But again, it's nice to have 6 computers running, or 5 computers running, or 4 computers running, as the case may be, at a time. And then basically you're leveraging lots of different compute to scale up, kind of mildly scale up pretty quickly what you're trying to accomplish. So in this case, I'll select a medium, 4 CPUs, 32 gigabytes. I'm going to put it into I'll put it into here's my project, the one we just created, and it's going to tell me here's how many allocation units I've got, here's how many CPUs I have left in my maximum, and here's how much memory I have left. And I'm just going to launch that, and it goes, and it'll actually take all of that software that we've developed in that image and put it onto hardware, put it all together, and then hand that over to you as a computer that you can access. You have total control over that computer until you tell us that you don't want it anymore, and then it gets torn apart, the hardware gets used by other researchers, and the image always stays the same. All right, so that's a very brief overview. Again, there's lots of documentation about Atmosphere. In the sake of time, I'm going to go ahead and get into doing the assembly really quickly. So I've already, of course, set up one of these virtual machines, one of these instances that is running. It's got our Trinity and Trinity image on it, all the software that's in there. And for this particular image, you actually have 2 options for accessing. You can access it through the terminal, which I'll show you, or you can actually open up a desktop. Now, I will remind you, this is a computer somewhere else. This is your cloud computer. And so I've actually installed the Atmosphere logo with the CyVerse logo as a backdrop to remind myself and hopefully other researchers that that is not actually local. If I go in here to home or to file system, none of the files that is on this computer are on my local computer. So here's my desktop in Tucson. Here's my desktop in the cloud, Tucson cloud, right? If I actually open up that file system, there's a bunch of stuff in here, but none of that is the desktop on the computer that I'm talking to you on, or that I run day to day. Now, to transfer data from the data store is very, very easy. I'm going to show you the command line way of doing it. So I can actually open up a terminal here, and I'm going to connect into the data store. So I'm going to use iCommands. iCommands is the most powerful and the fastest way to move data.
You
And
may need to
by
bump
learning,
the font up.
bump the font up. Excellent, thank you. I'll do full screen too. Why not? And let's hope that that's also bump. Oops, that's not the shortcut. You. I'm not sure how to do that on a Linux.
Then just go ahead.
Okay. And so what I'm going to do is connect into the data store to answer Randy's question. So I'm going to say init. So what I'm going to say, what I'm telling the cloud computer is go and ask and connect to my data store. So I'm going to enter, because you've actually already connected, you signed into Atmosphere and you started this, it already knows that I'm Blake Joyce, I'm this researcher, here's my username and all this other stuff. So all you have to do is put in your password, which is super nice. This will be your CyVerse password. So I'm going to type that in. Don't be scared. It won't actually show anything. That's for security purposes, so you guys can't see all my passwords and whatnot, or people looking over your shoulders. And so now I'm actually connected into my data store. If I type ILS, that is to say I commands list. So talking to the data store, it's always going to be I something, I command something. And it will actually list out all of the folders and all of files that exist inside of my data store. Just to prove that to you, here is that original Jove non-model folder. And here it is in my data tab. Here's my Jove non-model data tab. They are the same thing. They're talking to each other. This is just a different way and a much faster way to connect in. To get something from there, let's say I'm going to take the reads left and reads right. So I'm going to say I get, that is to get something from the data store and bring it to this computer. I'm going to say reads.left.fq.gz. So really what I'm doing is I want to pull that file. And that's actually a pretty big file, and it's already done. And I can see that on my local, here's the desktop on my cloud computer, and here's that file. It's already transferred. Let's actually see how big that file was. Here's the reads. Oh, they're actually pretty small. These are the example reads. But say I wanted to pull, I don't know. Yeah. Why not? Let's try this 43 gigabyte one, and we'll just see how long it takes. So I'm going to take this. Oh. I'm going to copy that because that's a long name. And I'm going to type I get, put that in there. I'm going to paste that in there. We'll just execute that as I talk, because I'm going to talk for a second or two. So basically what this is doing is going to the data store, grabbing those 43 gigabytes and transferring it over to the Atmosphere instance. Normally to transfer 43 gigabytes would take hours, if not much longer, depending on your connection and otherwise. So we're going to let that run and see how long that takes. Now, while that's going, I've actually got all the data set up inside of this. So we are going to switch over to the assembly. And I also, exit full screen. Okay. So again, we're on our cloud computer. I'm just going to minimize that because I can set it and forget it. And it's going to transfer that at some point. We also have, we used to support iDrop. That actually is no longer supported, though it may still work. I haven't actually tested this recently. These days we use Cyberduck, and we have links and documentations for setting up Cyberduck. So if you don't want to use the command line, you can actually transfer data between the data store using Cyberduck. All right, so without further ado, we are going to actually run the assembly, and it may be that that's as far as we get. So I will also mention that inside of the documentation, inside the wiki, I have all of the bash commands that you'll need to run at least the example data. And I'll point out where you will have to change. I highlight, bold, and underline it in the documentation, and I'll talk about it really quickly before I execute. So I'm going to open up. Ooh, come on now. All right, here's a new terminal. My old terminal's up there, and it is still running. Here's my new terminal. So I'm going to move around inside of this to execute. So we call that changing directories. So it'll be CD is the command. So ls lists everything that are inside of that computer, inside that cloud computer. If we go CD / and ls, so we've changed our directory to be the highest level directory that's inside of this computer. You'll notice that there's a directory here called tools. So CD tools. I'm going to see what's inside of tools. And you'll notice that we've installed all kinds of fun things. Blast, Bowtie, Expat, Hammer, Mongoose, RNA Hammer, Signal P, et cetera, et cetera, et cetera. There's also Transdecoder, Trinity Assembler, and Trinity Annotation. So because these are already installed, you don't have to actually bother with it. We've already done that work for you. We're going to be running Trinity, which is here. So we're going to go back to our desktop. by just typing cd. So, and I'm going to copy and paste the commands out of the documentation, and I'm going to put them here to run the example data. Now, you'll have to change some things because your data is going to be different than the data I have here. Again, this is just going to be experimental data that they include, example data that they, the Trinity folks include inside of Atmosphere. So we are on, so just to go back, here's our cloud computer. Here's a lot of information about it. One of the things to remember for Trinity is it's going to ask us how many cores we have. In this case, because I selected medium, we have 4 cores. You may also consider selecting large, in which case you would have 8 cores, and you'll have to change things just slightly. But I'm going to copy, I have both of those actually set up inside of the documentation. I'm going to paste, so I'm going to right click and paste that. Oops. Copy, I guess I didn't copy properly. And I'm going to paste that right in there. That's obnoxious. Sorry about that. So I'm not sure why that's not working, but I will swing back and try to figure that out. I'm just going to type it. It's just as easy doing it this way as it would have been. And I'm going to walk you guys through each of these. So what we may consider doing is I'll get you through this first step. I'll talk to you about some of the things that are going to occur. And then you can go home, and in the next couple of weeks, you can actually run the same thing, try to troubleshoot it and work through it. And then we'll go on to the next step. And this will be sort of like a kind of a systematic walking through an entire workflow. I had to do a lot of explanation, so regrettably this has gone a little slower than I originally expected, but it's good to have questions. I'm glad that we have questions. So what we have here is we're going to tell the computer, run Trinity. This is the sequence type, it's FASTQ. Here's the left reads. This is the file called reads1 FASTQ files. So that's an actual read set. Those are actual sequences in FASTQ format. And this is going to be something very similar to what you would get from a sequencer. Because this is a paired in, we're going to tell it the right reads here. And we're going to tell it, a little typo. It's always good to check for typos. So there's the right reads. And here's how many CPUs I have. And here's the max memory that I have. And that's going to have a G on the very end. And this is the command that Trinity gives you to actually execute this. Now, I'm going to copy this. Just for goodness sake. What's also nice, I will mention that if you press up and down, you can actually rerun old commands. And so you actually don't really have to copy and paste as much as I originally thought. So I'm going to move into the Trinity folder real quick and just show you one that I've already executed in the sake of time. So I'm going to go back to the tools folder. I'm going to go into the Trinity, CD, Trinity. You wouldn't have to do this as a user. What are the hyphens? The hyphens, excellent. So when you're moving around in the directory, these the backslashes, these things here, the forward slashes, are different directories. And it is analogous to moving around inside of your Windows Explorer or your Explorer files. So if you actually open the file system, what I've done is I've gone into this file and I've gone to Tools and I've gone to Trinity right here. So it's the exact same thing. In the terminal world, those backslashes, the hyphens. Oh, those are just how they were separating. That's how the Trinity folks separated out the versions of Trinitate and Trinity and other things from the actual names. So in this case, hyphens are fine. Hyphens, underscores are fine for spaces. Am I answering is it hyphens that you're asking about, or is it the, Yeah, again, spaces inside of the terminal they're not acceptable. It will not allow you to do things. It'll break stuff. So people use that or underscore to denote spaces. Hopefully I've answered that question. Anyway, so we're going to look inside of this Trinity folder in this exact same way. And I've actually run one of the sample data here under test. It was test Trinity Assembly. Yes. Okay. So here's all of our reads, and here's here's the reads that we're talking about here. Reads right and reads left. So I'm going to actually move into that folder just to show you again, because I've already run this, so I just want to show you what it looks like. Oh. Let me see, dash 8.1. Sample data. Test. Excellent. Okay, so we are now in the same place as I have navigated to here. So inside of there, I'm actually going to execute that. And let me move this up. Sorry, it's getting a little out of view. We're going to execute that command I typed in before. I'm just pressing up to go back to my old. Oh, well, for some reason it didn't save it. That's fine. FQ left reads dot underscore dot left dot FQ. So that's actually going to tell it use this file. That's my left file reads. Here's the right reads. So we're going to do reads dot right dot FQ. And then we're going to tell it that we have 4 CPUs because that's how big the cloud computer is. And we're going to tell it that we have a max of 30 gigabytes of memory because that's how much we have. So we're just going to run that. Cross our fingers. I'm going to type in my password again for my CyVerse password again. Oop, I'm going to do it right. So the data set that Trinity provides is very, very small. Trinity, the assembly, often takes days or possibly weeks, depending on how big and how much data you're trying to put together, how many reads and how you're trying to put together. And so what's basically happened really quickly is that we ran Trinity, and it will actually output this as it goes along over the course of hours or days or however long it takes. And it'll say Trinity phase 1. We're going to cluster all the RNA-seq reads together. We're going to convert input files and do all kinds of stuff. Jellyfish is going to build your k-mers and start to build graphs. It puts together all those and actually starts to do everything. And then when it's finally done, it will say that it writes out. And here is where it is: tools, Trinity, sample data set, test Trinity assembly, Trinity out directory. So when you run Trinity, it creates its own out directory, so it's very easy to find all of the outputs. And then this will be your assembly in faster format. Okay. So again, when you run data, especially lots of reads, you will have it run for days, but it will output, and that'll be your final assembly there. And you can actually find that in, again, a number of different ways. But we're going to go, this is the same as moving into it. This is just nicer looking. We're going to go in here, and here's all of the outputs. Trinity does a lot of log outputs, and it checks your reads, and it does all kinds of fun things. This is your final assembly. We're going to try to open that. And voila, here is Trinity. Here's the name of the config, isoform 1. Here's the length, and there's the standard fast format, and then here's the actual config. There. Yes, Meg, that's an excellent question. So the one thing I would absolutely suggest is I'm going to show you how to put this back into the data store, and then I like to rename it. And again, no spaces in the name, but we're going to rename it. So we're going to do all of that in the last few minutes here. Great question. So I'm going to close that, so you can actually get your assemblies this way. We're going to close this, and I'm going to put this assembly. I'm only going to put the assembly back into the data store because that's all I'm interested in right now. Next time, it looks like it'll have to be next time, I'll show you how to output counts tables and differential gene expression and do assessments of the FASTA files and all the fun next steps. But oftentimes, getting the assembly is sort of the first big hurdle to get through. So I'm gonna put this file back into my data store because it's safe inside of that data store. If the cloud computer goes away, anything stored on this cloud computer will also go away. So I like to put everything back into the data store, and then I finally consider that complete. Okay. So here's how we're going to do it. We're going to go back to our terminal. This is the one we just ran. I'm going to do list ls to see all of this files. I'm going to move to the Trinity out directory. So I'm going to change my directory, cd, to Trinity_out directory. I'm going to list all the files again so I can see, here's all these files that we were looking at before, and here's my assembly. That's the one we really want, is right here. Is it worth reassembling data using the new version of Trinity, or do you think it would just be confusing stuff? It can depend. Usually the new versions won't create new types of assemblies unless you give it new parameters, but it's always good to check that. And so that's part of the experimentation I was talking about before. I personally like to run it on the newest version and one version older, and then just compare and assess to make sure they're not strikingly different. If they are strikingly different, I go with the newer version. Again, it's your science. You can decide what kind of time inputs you have. Again, I will show you guys how to do the assessment in the next installation. So just to finish this up, and hopefully everyone can see this, I'm going to put that FASTA file into my data store. So I'm going to say I put, because I'm going to take the file from this computer I'm working on, the cloud computer, and put it into the data store. So it's called icommands put, or I put. I'm going to say I put Trinity.FASTA. And I'm just going to return that. And it now sends that FASTA over to my data store in the discovery environment. So if we can actually see that, I'm going to go, I'm going to do a refresh, like so. And I'm going to go down to the bottom here, and voila, there's the Trinity. We just put it in there at 11 o'clock on the dot. All right, that's pretty awesome. And it is 166 kilobytes. Again, because this is example data, it is much, much smaller than what you'll have. But there it is, 166 kilobyte assembly. That's all of our FASTA. And next time, I can show you tools to assess that assembly and turn it into protein peptide sequences, into a GTF, into a GFF, or otherwise. Can you download and install temporarily additional software to your computer? Yes, absolutely. So again, one of the cool things about cloud computing is this is your computer until you tell us you don't want it anymore. So I can install, I can update, I can do anything I want. Just remember that anything I do, once I get rid of this computer, it goes away, unless I image this computer. So I can actually take this, I can install updates, I can, let's see, sudo apt-get is a way to update. I think it's upgrade for apt-get. And I'm now going to go and it's going and finding all of the new software since we installed all this stuff. And it's going to say, do I want to install that? Sure. And it's going to run away and do that probably for a while. Now, I will also mention in the 15 or so minutes, we transferred that 63 gigabyte file in only just a few minutes. It hasn't been probably 20 or so minutes. And I can actually see that file here, which is, again, really, really incredibly quickly. And we were actually doing computation even while that was running. And so that's sort of the power of having some of this, the cyber infrastructure in place. Once you get it into the system, you can really work with your data very quickly instead of having to upload, download, large data files, which can really eat up a lot of your precious time. And so I'd prefer to spend that precious time doing experiments like what Meg was asking, trying assemblies in different ways, trying assemblies on different versions, using entirely different assemblers. And I will show you how that assessment kind of looks. In fact, let's just do it real quick, and then I'll let all of you guys go. So in this same outputs, I have the assembly here. I already ran that. And I've used RNA-QoS, which is a tool for assessing assemblies. And what we actually did, Apindra and I, way back, is we ran 3 different assemblers on the exact same read using standard settings. And if we look at this PDF report really quickly, pop-up blockers, what RNA-QoS will do, amongst other things, is it will give you summary statistics. And we use IDBA, we use Trinity, and we use Spades, which is another assembler. These are 3 different assemblers. And you can very quickly take those assemblies from the different assemblers, and you can look down and see, is my final assembly that different if I use different versions of the same assembler or different assemblers altogether or otherwise? And now you can actually assess and pick the best, in your mind, assemblies for your science. And that makes me rest a little easier at night. I like these sort of things, and I like to be able to prove to my reviewers I've done my due diligence, and I've actually looked at many different assemblies, and I looked at every step, and I did the best I could to get the best data for asking questions and doing experiments further down the line. That is the differential gene expression, the annotations, metabolic profiling, etc. Does Atmos send complete notifications? Can you write a bash script to copy results at the end? Yes. Atmosphere does not send completion notifications because it's a separate computer. I'm working with the Atmo team right now I'm glad you've asked this question, Ken. One of the things I would really love to do, and they're actually working on this for one of our updates, is here under activities, when you actually have a bunch of these running, it would show you either a graph or summary statistics every couple minutes of how much computation or whether that computer is actually running. So let's say I fire this off and that assembly is going to take 3 or 4 days because I have a really big data set. This would basically report out to me. I could very easily come here, look at that, and say, oh yes, my assembly is still running. Or, oh, my assembly completed because it's all the CPU and the memory has flatlined. So that's actually coming a little further down, a couple months from now, probably, or towards the end of the year. That particular functionality has been put into our production pipeline. So it doesn't send completion notifications. The discovery environment absolutely does. You'll get notifications here. Here's tools that I've been running and trying to break and doing other things. You'll get it's submitted, it's running, it's succeeded, it's failed, and it will actually send you the discovery environment will actually send you emails as well if you'd like it to. You can also turn that functionality off. Some people just don't like to get email reminders. And as for executing the bash script at the end, yes, absolutely. If you have the ability to write wrappers, you can totally do that. And that becomes very interesting. And just very quickly, what I'll show you is when you actually make new instances, one of the clever things you can do, if you have that ability, again, we like to speak to all levels, beginners, intermediate, and advanced users. And so when you actually are creating these, what I didn't show you guys before is down here in advanced options, creating the instances in advanced options, you can actually upload bash scripts so that when the computer is done putting together all the software and all of the hardware and your cloud computer is ready to go, it will actually execute a bash script right here if you add that script. And we have a bunch of them in-house that we've developed. So what I've seen, some very clever people, what I've seen them do is, especially for Trinity, because it needs to pull in databases, they'll actually write a bash script and execute that database pull into the cloud computer, so that when you actually go into the cloud computer and start doing work, you've already moved those files and it's done, and you're just ready to go. It's just sight unseen. So great questions. Great questions all around. Are there any more questions? So I've shown you the assembly bit.
Blake, as
Yes.
as you're doing the wrap up here, would you do me a favor and go to the slide?
Yes, I'm working on going back to. Yes, absolutely.
Thank you.
Thank you for reminding me. There we are. So just wrapping up real quick. Please send me emails. I will go ahead and answer emails as well. Or answer your questions for as long as I can. I will have to run here in about 15 minutes, but I'll stick around and answer questions as best I can. I will also remind you guys, though, that we are constantly upgrading, updating, modifying, improving. So if there's something that you didn't see today that you'd love to see, or if there's some questions you have, please don't hesitate to reach out to me. But bioinformatics is always a moving target. There's lots of tools out there. They're constantly, the new versions are coming out. And so we spend a lot of our days trying to keep up with all of the different possibilities. And because everyone's science is a little bit different, you actually wind up with a kind of an infinite number of possibilities as far as tools, analyses, possibilities within any given tool or workflow. And so what I'd like to point out is that you, as users, can actually integrate any of these tools or develop any of these things to fit your needs. And we'd be happy to show you how to do that, because we're really here to help you do what you need to do, and give you the power to actually get to that goal. And that really is what we're trying to get people to. And that's why I, as a user and as an employee, have really enjoyed CyVerse, because I was a user for a very long time before they hired me. So we are, of course, updating the Trinity Assembler. That will also be available in the DE. They're up to version 3 now, if I remember correctly. What's nice about this new version is it runs much faster, I'm told. And they have some summary statistics on the Trinity website to kind of prove to that effect. And of course, one of the major pushes we have for the next 6 or so months is updating a lot of the downstream annotation tools. There's a group of people working with us here at CyVerse to basically start to answer this question of, once we make it to annotation, which can be its own hurdles, how do we actually sit down and make biological sense out of this? How do we do metabolic identification or flows or network analysis? Or how do we move out to other platforms that do this really well? So one of the things that we really love to do here at CyVerse is we like to reach out to community members that are doing their own analyses elsewhere, that are creating their own platforms, and basically be able to interoperate with them and create ecosystems of analytical tools, because those ecosystems are much more powerful than any one thing that we can do here, even though we're constantly developing. It makes more sense to work with community members, because people in the invertebrate biology world are going to have much different needs than people in the exotic plant world, or in the fungal pathogen world, et cetera, et cetera. So that's really where we like to be, is speaking to that level of users and then helping communities build tools so that they can deliver those tools to their communities. Because we're domain scientists, but we don't know every domain, right? You know your domain. We can help you speak to your domain. So that's a huge push, and we have a lot of tools already available, but we really would like to formalize and sort of do sort of get to a place where we're doing meta analyses and otherwise. I see Sophie's asked a question: is it a good idea to combine the transcriptomes of various assemblers to create a better transcriptome? Excellent, excellent question, Sophie, and I will speak to that here in just a second. I will go ahead and just wrap up and say that we're of course funded by the National Science Foundation grant. We've had 2 5-year grants. We're currently in the 9th year of the second grant, or 9th year overall, which is the 4th year of the second grant. And we're really appreciative of the National Science Foundation allowing us to help you guys. And hopefully this has been useful. I will continue to answer questions as best I can. And yeah, please don't hesitate to reach out to us. And if you would like to see the rest of the workflow, I will schedule a time next month and we will go through the next steps. This video will of course be available to you on YouTube, And if you can't make the next time, that video will also be recorded, and that will be available to you on YouTube as well. Thank you for your time, and I will take questions for at least the next 10 minutes before I have to run off.
Okay, let me jump in and let everyone know that I will get with Blake and get an email out to everyone with the date of that next webinar as soon as he and I can get past our next meetings today. I will also send out a follow-up email today so that we can so that you have links to the information that was covered, and so you can provide us with any feedback for improvements for the next webinar. Okay. If there are no additional questions for Blake, I will go ahead.
Yeah, sorry. Let me also go ahead and post the, I'm going to end the slideshow, and I will post the link to the documentation. I should have done that immediately, and I apologize. Here's the documentation, and of course, we will send out, you'll notice that there are the command lines that I've run. There's grave details about each of the steps. They're set, of course, in a protocol. This will continually evolve as our abilities also evolve. And let me go ahead and link this to you guys here. So if you would like to go ahead and do the assembly and get ready for the next webinar, which will be assessment, annotation, differential gene expression. And I apologize again for not getting there, but there's just a lot to do. All the documentation is going to be there. And if you'd like to go ahead and just run through it and then randomly ask me questions, my email is bjoyce3@CyVerse.org, and I am highly discoverable.
Actually, if I can suggest something different, please send questions to support@CyVerse.org, not directly to individuals. And I ask that users do that so that everyone within CyVerse can see the questions and know what's going on.
Okay, that makes sense.
Thank you.
All right. Well, again, I will stick around for 5 or 10 more minutes if anyone has questions or specific questions about what they want to do. And thank you again for your time.
Blake, I need to end the recording. So we're actually gonna, and that's gonna end the chat. So,
oh, I see.
Okay. So I think people, you know, it's been over an hour. So people can send follow-up questions into support if they need to. But for now, for today, let's call this a wrap.
All right.
Okay. Thanks, everybody. And we'll let you know when the next webinar is going to take place as soon as we get the dates set. Bye-bye.